results, what was it? What was the compound? And, you know, of course, I love puzzles. This was perfect for me. And so after I finished my undergraduate and went to be a graduate student, I went to work for him. But I ended up not working for him. I worked in his lab. But there was actually a postdoc in the lab, a really great guy. He's shown here. His name is Kazu Kurosawa. He was Japanese. And this picture was taken a year ago. I left Sheffield, well, he left Sheffield actually before I did. So he left Sheffield in 1966. And for a long time we would communicate, and then we lost touch. And I decided it was time I really got back together with him. And so I made a plan, and I found him during a trip to Japan last year. And we had a wonderful time. We really had a good time together. But he taught me how to play Go, taught me how to play Japanese chess, shogi. And he was, again, someone who was really interested in puzzles, and he was a great teacher, wonderful teacher. He was one of these people who would say to you, oh, you should do this experiment, and you do it and it would work. But he would explain it in such a way that by the time you'd finished the experiment, not only did it work, but you knew why you'd done it. And so the next time you had the same sort of thing to do, you knew exactly what to do. And I'd not come across a teacher or mentor who was quite as good as him. He was fantastic. And this, of course, is Go. Love Go. Though I was very disappointed to hear there are now machines that can beat people at Go. That's not so good. But the thing that really happened while I was a graduate student in Sheffield was that I got all the results I needed for my PhD thesis in the first year. So 1 year, and this was because of Kazu, okay? He told me what to do, I did it. I found this one compound that my professor had predicted should be present as part of a biosynthetic pathway. And many other people had looked for it, none of them found it. I found it. It was in the tree that I was studying. And so I got all the results for my thesis, but in England you have to spend 3 years doing a PhD. You can't drop out and do it early. And so I ended up with 2 years really to do whatever I want. And the professor was rather nice and let me do what I want because I'd already got all the thing. And I went over to the library and I used to do a lot of reading, and I came across a subject called molecular biology. And that was as a result of reading this book called The Thread of Life by John Kendrew. John Kendrew was a Nobel Prize winner. He got the prize many years ago, actually, at the same time that Jim Watson and Francis Crick got the prize for the double helix. And Kendrew got it for the first protein structure. But this book described the early days of molecular biology, what it was like to suddenly discover that there was all of this stuff going on inside cells that could be explained in terms of chemistry. And that's what I became, and that's what I am today. So this was something that played a hugely important part. And I think all of you who are here at the university, you must take advantage of all of the things that a university can offer you. So it's no good just going to classes and taking exams and passing exams and so on. Go and look around you and see what else is available. Go to the library and read other stuff. Go to lectures in subjects that are far removed from the ones you're in. Because you simply never know when you're going to find something that is so fascinating, you say, gosh, that's what I want to do with my life. And I have another lesson that comes from that, is that when you do find these things that you can be really passionate about, and even though they're not what you're supposed to be doing, they're not what the subject that you're thinking, if you find something you're passionate about, drop what you're doing and follow your passion. Because this way you will be successful, you'll be happy, and you will have a good life. And it won't matter. You can do this at almost any stage during the course of your career. So don't be afraid to switch. If something looks more exciting than what you're doing at the moment, do that. So when I finished at Sheffield, I went looking for a postdoc. And instead of being a chemist, I wanted to be a molecular biologist. And so I was changing fields completely. So I applied to a bunch of labs, and they all turned me down, except for this man, Jack Strominger. And he was at Wisconsin at the time, at Madison, Wisconsin, and he offered me a position, and I later learned he offered me a position because I was English. He had a very interesting approach to hiring postdocs. He would hire someone from a country, and he thought if that person turned out to be good, he'd hire the next person from that country who applied and would continue doing this until he got a bad one. Okay. And then he would pick another country. And I was lucky, he had an Englishman who had worked with him for a long time, and so just the fact that I was English, I think, was the main reason he picked me. Anyway, he was terrific. But about 2 months before I was scheduled to go to Wisconsin, he sent me a letter. You know, people used to send letters and things in those days. Now it would have been an email. And he said, can you delay coming for a couple of months because I'm just moving to Harvard. And so quite by chance, I ended up going to Harvard instead of going to Wisconsin. And that turned out to be lucky. But what was even luckier was that Jack Strominger, who'd worked for 20 years on bacterial cell wall biosynthesis and had worked out how bacteria made their cell walls, had decided he wanted to switch fields, and he wanted to work on HLA antigens and do something eukaryotic. And there were just a few bacterial projects left, and I ended up working on one of them, but he wasn't really interested, and so he sort of left me alone and let me do what I wanted to do. And that's how I like to work. I don't like people telling me what to do. In fact, I can tell you almost as soon as someone says to me, do this, I want to do the opposite. I think, you know, well, if they thought that was worth doing, why don't they do it, you know? And when someone tells me don't do something, then I know there's something really interesting. I've always found interesting stuff away from what people think I should be doing. So anyway, while I was in Jack's lab, the most important thing that happened was that I was put on this project to sequence a transfer RNA. And this was in the days when there were no really fantastic methods for sequencing RNA. And there were no methods at all for sequencing DNA. And so there was an old-fashioned method, if you like, using unlabeled tRNA and doing chromatography and all sorts of things that a guy called Bob Holly, who ended up being a Nobel Prize winner, worked out. But there was a new method that was just being developed by Fred Sanger. And he was at the MRC lab in Cambridge. And I read what he'd been doing, and I thought this would be perfect for my project. And so I wrote to him and said, can I come and learn the techniques in your lab? I went over, I spent 6 weeks in Cambridge, learned how to do it, came back to Harvard and set up the exact same apparatus and everything that was needed. And I ended up being the first person in the Boston area to be sequencing RNA using Fred Sanger's method. And that worked out beautifully because a lot of people came to me to learn how to sequence RNA, including Wally Gilbert, who later went on to get the prize for sequencing DNA, and a bunch of other people. And so that was very nice. Go to the next one. So this was Fred Sanger, shown down here. Oh, sorry. So this is Fred Sanger here, this is Wally Gilbert. I visited Fred's lab to learn RNA sequencing in '69 and came back, and Jack Strominger had a lot of money, and so he paid to put in all this equipment, and he didn't really know what most of it was about, but he let me have my head and do what I wanted to do, and so that worked out very nicely. So after the time at Harvard, it was time to try and get a job. And at the time I was married, I had 2 children, and so I needed a job. I couldn't just be a postdoc forever. And so a friend of mine told me that there was the perfect job for me at Edinburgh University back in the UK. And I'd always thought I would go back to the UK, and so I applied for the job. And I waited, and I never heard anything from them. I didn't even get a note saying, you know, thank you for your application or anything. And then, in the meantime, I got offered a job at Harvard, and I got offered a job at Cold Spring Harbor. But I really wanted to go to England, but what could I do? You know, I had nothing. And so I went for an interview with Jim Watson, and I spent 10 minutes in his office, and he offered me the job. He offered me a job at Cold Spring Harbor, and I was just leaving, and he said, oh, and by the way, you probably should come down and visit us, but the job is yours. You know, I don't care what other people say, the job is yours. And what he wanted was to have someone who could sequence SV40 DNA. This was a relatively small DNA virus, and the way that people would try to sequence DNA in those days was by making copies into RNA and then using the RNA techniques that I'd learned in Fred Sanger's lab and that I was good at. But as soon as I got down to Cold Spring Harbor, I realized that there were 2 other groups who were sequencing SV40. So it seemed kind of stupid to be the 3rd group doing the same old thing. And so I looked around for some other things to do. And I came across these things called restriction enzymes. And what had happened, I went to a lecture at Harvard Medical School by Dan Nathans, this fellow up here. And he gave this wonderful talk indicating how you could take SV40 DNA and cut it with this enzyme, the one enzyme that was available at the time called endonuclease R, and you could get nice fragments. And among those fragments, there were some small ones. And one of the reasons that RNA sequencing was developed before DNA sequencing was that there were no small DNA molecules to practice on. RNAs, there are plenty of small RNAs, proteins, there are plenty of small proteins, but DNA, even the smallest DNA, was 4,000 or 5,000 base pairs long. And you can't really develop methodology very well if the thing you're looking at is so big that you don't have a second way of determining whether your techniques are correct or not. And so I thought this was the perfect way to make some small DNA fragments with which to develop DNA sequencing techniques. And so I went down to Cold Spring Harbor and told Jim that this was what Jim Watson, that this was what I wanted to do. And I said I'm not interested in sequencing SV40 because there are 2 other groups doing it. He got really pissed off. We fell out right there and then, and I think until a few years ago we never really made up. So we were always fighting with one another the whole time I was at Cold Spring Harbor. Anyway, so we started doing this restriction enzyme. We made endonuclease R, we made several others, and it soon became clear to me, not to other people, but somehow to me, that these enzymes that we were making, there were loads of them. There weren't just 1 or 2, there were a lot of them. And so we started looking in various bacteria that I brought with me from Harvard. These were bacteria that I brought with me because I thought I might continue doing some cell wall biosynthesis work, if I had some spare time. And so we opened these up, and every single one of them that we opened up had a new restriction enzyme in it, and one with a specificity that was different from the ones we already had. And so what happened was that I ended up focusing my research at that time on finding new restriction enzymes. And these enzymes were really cool. You could take a big piece of DNA and cut it into fragments, and there were all sorts of things you could do with those fragments, including clone them. And so these enzymes became one of the foundations of the biotechnology industry, and everybody wanted them. And I had a collection of about 30 down at Cold Spring Harbor that we discovered. These were not available anywhere else. No one else had them. And so people used to come to Cold Spring Harbor to meetings, and they'd bring tubes of DNA with them, and they'd slip out of the meeting, come down to my lab and say, oh, do you have an enzyme that will cut this? And so we got set up, and sure enough, we could always find things that they were interested in. And it occurred to me that you could make a business out of this. That, here was something that would be really useful. And so I go and I talk to Jim Watson, and I say, look, why don't we set up a company at Cold Spring Harbor with a Cold Spring Harbor name, and we'll use the profits to support research. We'll just put the profits back into Cold Spring Harbor, and we'll use it to support research. And he basically told me I was crazy, he couldn't make money doing this, he's not a particularly good businessman, and he wasn't interested. And he also said, well, it's kind of dirty to be involved in industry. This was a time when, true academics didn't get tied up with companies. So I looked around, I knew there was a good idea here, and so I looked around, and someone down at NIH told me, well, there's a company in Massachusetts that is going to be selling these soon, and he put me in touch with a person at Miles Laboratories, and the person I talked to said, well, it's not us, it's this fellow over in Beverly. His name is Don Cohn, you should go talk to him. And that was really how New England Biolabs got started, because it turned out that Don Cohn had exactly the same idea I did. He wanted to make these enzymes, sell them to the research community, and instead of becoming personally wealthy, he wanted to reinvest this money back in research. I'll tell you a little more about Biolabs and what happened later on. one of the things that we were doing with the restriction enzymes, in addition to just finding new ones, was to start to map some of the DNA molecules, and there were a lot of people interested in adenovirus at New England Biolabs. And so we started with Phil Sharp, and so Phil Sharp up here. We ended up having a paper together mapping adenovirus DNA. And in 1974, I got a postdoc from Harvard, Richard Gelinas, shown down here, and we were looking for a project to do, and we thought it would be fun to see if the promoters, the regions on adenovirus that told the RNA polymerase where to start transcription, see if those promoters were the same as the ones in bacteria. Because Wally Gilbert and Mark Potashny and others had just begun to characterize promoters in bacteria. And everybody assumed, whatever was true for a bacterium was the same true for all eukaryotes. But we just wanted to test it, and we thought this would be a good use of both the restriction enzymes to label and isolate fragments of adeno-DNA, and then to map where the transcripts began. And so we started doing this, and Richard was the postdoc in my lab who got this started. And we had what I thought was a very good idea. I usually use this slide just to talk about Jim Watson. So this person here, Norton Zinder, is probably the only real friend that Jim Watson ever had. Jim is a difficult man, was a difficult man. He made an awful lot of enemies, but for some reason he and Norton got on really well. And I met Norton because he used to come out to Cold Spring Harbor, and he and I got on really well. And I would say every month or a couple of months or so, Jim had found some new reason why he wanted to fire me, because I wasn't doing what he wanted me to do. And Norton was the person who would go to Jim and say, why don't you back off? Richard's okay, let him go. And so Jim would then back off and things would be okay for a few months. So Norton played a very important part in my life during this period. So we decided, Richard Gelinas and I, that we wanted to map messenger RNAs onto the adenovirus genome. And it was at a time when we didn't know about caps on messenger RNA. We thought they were just had 5' triphosphates in exactly the same way that bacteria do. And so a typical messenger RNA would have a 5' triphosphate, and it would have poly-A at the 3' end. Poly-A had been discovered at that time. And the idea was that if you treated that with ribonuclease T1, which is an enzyme, an RNase that cuts after every G residue, you would end up with a series of oligonucleotides that would look like this. All the ones in the middle would just have some string of nucleotides running to G. The ones at the 3' end would have the poly-A on them, and the ones at the 5' end, the ones that were the start point for transcription, would have this triphosphate. And so I came up with what I thought was a rather clever way to find everything containing these triphosphate ends. And it was a very simple experiment, really. You could take the mixture of RNAs, you could run them down a poly-A column, and that would then get rid of all of these with poly-A on. And now there was something special about the triphosphate, and we thought we could capture that. But just as we got going on these experiments, caps were discovered. So this turned out to be absolutely key for our experiments, because all of a sudden now there was an even better handle that would allow you to find the 5' ends. And that's because these cap structures have a glycol residue here, 2,3-cis-diol, and that will bind to columns that contain borate. And so what we could do was to capture these just by binding them to a borate column, and then we thought we could fingerprint all of these, and we will see the start points for all the messenger RNAs. It's sort of like an RNA-seq experiment that you do these days, but except this was done on something much simpler. So here was the idea: treat it with ribonuclease T1, and then capture not the triphosphates, but capture these caps. And so we did that, and we did that with late adenovirus RNAs, where we knew there were about 20 different messenger RNAs that were present. Okay? And so we expected to see 20 spots on this two-dimensional fingerprint. This is a two-dimensional fingerprint. And instead of 20, there were just 2. And it turned out that this one was just the cyclic phosphate version of this one. And if you look late, then you see that there's just 1. And so here we've got 20 messenger RNAs in adenovirus, and they all had exactly the same 5' T1 oligonucleotide. And so, of course, you know, the first thing you do when you have a student who comes and tells you about this, and that the experiment failed because I knew this wasn't what we were looking for, I said, well, you know, go back and do it again. And so he went back and he did it again and got the same result. And I said, Richard, you know, sit down, let me do it, I'll show you how to do it. So I did the experiment and got the same result. And I learned something really important here. So one is not only that graduate students, postdocs are sometimes okay, but that failure is something that is really good. When you do an experiment and it fails to produce the answer that you expect, this means one of 2 things. Either you screwed up when you were doing the experiment, maybe you dropped it on the floor, you spat in the tube or something, or nature is trying to tell you something. And so when things fail, it's always worth going on and trying to figure out why they failed. And so to this day, I love it when students come to me and say, well, you know, I did what you told me to do and it didn't work. I think, great, nature's trying to tell us something, let's find out what it is. I think this is something that students often don't appreciate, that failure is really important. People who never fail at things almost never learn anything. Failure is the best teaching device out there, and sometimes it can lead to important discoveries. So we spent about a year trying to figure out what was going on here. This was the first paper that came out, one predominant 5' undeconucleotide. It was 11 nucleotides long. And then we basically looked at these capped oligonucleotides and found something very unusual about them. First thing was that if you take the adenovirus genome and break it up into pieces with a restriction enzyme, and then hybridize all of the RNA, so you just pick up the messenger RNA that is present in each of these fragments, you got exactly just this one oligonucleotide every time. So it didn't matter where you hybridized, you always got this oligonucleotide. When pure RNA from several well-known proteins, there were 2 co-proteins in adenovirus, got exactly the same result. The same undeconucleotide was there. And so we slowly honed in on the fact that every messenger RNA had this thing that it's 5' end, but how could that be? Because, you know, there were so many different messenger RNAs. And we also discovered that if you take mRNA and hybridize it to the fragment that is coding for the protein, and then treat very mildly with a ribonuclease, you lost the cap. Cap disappeared completely, it wasn't there anymore. And it didn't matter where you did this experiment, you lost it. And so that was also a bit curious. And I have to say, this was a time when people were saying, you know, why don't you go and gym? Why don't you go and do something else? You're wasting your time here, you know, you're getting nowhere, you're just looking at some artifact, and people wouldn't believe it. But both Richard and I really knew that we were onto something big here. I was more convinced than he was. He was doing all the experiments, so, you know, it was easy for him to get discouraged. And we got into this habit. Every Saturday morning we sit and we talk about some new experiments and how we really do the key experiment this next week. And then a lady called Saida Zain, she was a Pakistani postdoc in my lab, she found and showed very clearly that this capped oligonucleotide was coded in the genome, but it wasn't coded next to the main body of the messenger RNA. And so that turned out to be an important finding. And then we discovered that early RNAs, so during adenovirus infection, when you first infect the cell, lots of early RNAs get made in very small quantities, and they're quite difficult to deal with. Late in infection, everything comes from the late promoter, and it is making the co-proteins and the things that adenovirus needs in order to make a new virus particle ready to go ahead and do something. And so we found that the early RNAs had a different oligonucleotide, and that wasn't lost by ribonuclease treatment. So one day, early in March in 1977, Richard and I were sitting in my office, and he was explaining some very complicated experiment that he wanted to do. And I was only half paying attention, and all of a sudden I got the idea, this is the experiment we need to do. And the experiment goes as follows. If you think about all of our results, well, here was the DNA, here's the RNA. We knew that at the 3' end of the RNA there was this poly A, and that wasn't coded on the DNA, and so that would stick out. And in fact, people, Ray White in particular, had done experiments showing that you could show where this poly A was by hybridizing poly DT to it, and then looking in the electron microscope, and you could see very clearly a marker showing you where that was. And we also knew that at the 5' end of the message, there was something, and it wasn't coded right next to the main part of the message, it was coded somewhere else. And so it too should be sticking up off the DNA. And we thought if we can find exactly where that is coded, we could do exactly the same experiment. In our case, we don't want to use oligo-DT, and what we want to use is something that is coding for this tail, the fragment of adenovirus. And now you can do something very nice, and you can make things called R-loops. So if you take a messenger RNA and hybridize it to DNA that has been denatured, it will form a DNA-RNA hybrid where the RNA is coded and the other strand just remains single-stranded DNA. And this is because DNA-RNA hybrids are more stable than DNA-DNA hybrids. And so this was the R-loop experiment that I thought about while Richard was talking about something else. And I said, sit down, this is the experiment we're going to do. And the experiment was simply to make an R-loop and to use a DNA probe in order to catch this 5' end. And I had a hypothesis about where this was coming from. I thought it was coming from an RNA that was coded by adenovirus that was known, it's called VA-RNA. We knew where it was coded, and I thought, well, what this is doing is priming synthesis of the messenger RNA. And so we'll use a fragment of the adenovirus genome that codes for this RNA, and we'll just stick it in and do the experiment, do the electron microscopy experiment. The only problem with that was neither Richard nor I were electron microscopists. But fortunately, just down the hall, we had two very good electron microscopists, Louise Chow and Tom Broker, and they were really expert electron microscopists. And so we went down on a Saturday morning and said, hey, we have this idea for an experiment, and what do you think? Do you think it can be done? And they looked and they said, well, no one's ever quite done that before, but yeah, we think it should work. And so we went away, Richard made the appropriate DNA fragment that we needed, and we already had the messenger RNA around. And so on the Monday morning or Tuesday morning, and we gave the reagents to Tom and Louise, they did the experiment, and lo and behold, they saw exactly this picture that we predicted, that there was going to be some DNA hybridized to the RNA. Except it was just a little bit more complicated, and there were actually two loops of the DNA. Because it turned out that this 5' end actually had 2 exons instead of just 1. We thought the molecule had just folded badly in the microscope when we first saw it, but Louise persisted, and by the end of the day she'd seen lots of these, they all looked the same. And that was the point at which RNA splicing was discovered. And this was, it was fantastic. We spent the day going around telling everybody about it. People were talking about Nobel Prizes and so on. Because here was something that was so fundamentally different between bacteria and eukaryotes, and it was something that had not been predicted. It came as a complete surprise to everybody. So this was the paper that we put out. It's called An Amazing Sequence Arrangement at the 5' ends of adenovirus 2 messenger RNA. We sent the paper into Cell, and there were 3 referees who looked at it, and they all said, well, they thought amazing was not an appropriate word to use in a title of a paper. Scientists don't do this sort of thing. And so I get on the phone, and Ben Lewin, who was the editor of Cell, said, why don't you find another word to describe this? And so I get on the phone with Ben, and we start talking, and after a little while I said, Ben, when you first saw this, what did you think? Oh, it was amazing. I said, I rest my case, and so he let us use it. So this was, to the best of my knowledge, the first time that the word amazing was ever used in the title of a paper. So once we had made this discovery, the question is, what are you going to do next? And so one of the things we really wanted to do was to work out exactly where the splice junctions were. And we did a bunch of experiments. We did some sequencing experiments. We sequenced the 5' end of the RNAs, so we knew where the little bits and pieces were. But it turned out that there was so much splicing going on in adenovirus that the only way to really get at the splice junction was to sequence the whole genome. We also tried to set up an in vitro system to study splicing, but we made the mistake, we picked the wrong promoter to do in vitro experiments with, and Tom Maniatis up at Harvard did a much better job, and they got great results. And so we ended up focusing on sequencing adenovirus, which we did. This was the second largest DNA that was ever sequenced. And as a result of doing that, I got interested in computation. Because what happens when you're doing sequencing is you get little fragments of sequence. You have to work out how they all get put together in order to make the complete sequence. And at the time, there were no programs available to do that. And so we wrote the very first assembler that anybody ever used to put sequences together. We did that with a guy over at Stony Brook. And so we also started to develop programs to analyze the sequences once they were made, and again, these were pioneering efforts. And then we began developing what I call bioinformatics. I always put bio in front because it's not about computation, it's really about biology. And I think a lot of people who do bioinformatics forget the importance of the biology here. So having done all of this, having gotten these assembly programs going, we didn't have a computer at Cold Spring Harbor. We were operating over at Stony Brook. And so I went to Jim and said, I think we need to get a computer here. And he looked at me and he said, over my dead body. He said, we are not having computers here at Cold Spring Harbor. And it turned out the reason he didn't want them was that he felt that if his secretary was using a computer to, write his letters and send them out, everybody would be hacking in and finding out what he was saying. He was just paranoid about this. Anyway, eventually we did get computers at Cold Spring Harbor, and when I left Cold Spring Harbor in 1992, he said he'd made two mistakes while I was at Cold Spring Harbor. One was not to start this company making restriction enzymes, because at this time Biolabs was doing really well, and in fact that was where I moved after Cold Spring Harbor, and he admitted that not bringing computers to Cold Spring Harbor when I first answered, that was a mistake too. Jim doesn't often say he made mistakes, but this was a good time. So we ended up, Phil Sharp and I, sharing the prize in physiology or medicine in 1993. That was wonderful. I've got to say, the Swedes throw the most incredible party you can imagine. So you go to Stockholm, you've got 10 days in which they feast you. And every day they take you to a lunch or they take you to a dinner that is so special that if that was the only thing that happened to you in the year, you would remember it and say, well, that was the highlight of the year. And imagine 20 of these things over 10 days. They're unbelievable, just incredible. I recommend it to all of you. So just as we were leaving Cold Spring Harbor, we made another nice discovery, and that was the discovery of base flipping. So we had been working with a fellow called Jiadong Cheng, who was a postdoc at the time at Cold Spring Harbor, working in another lab, and we'd been collaborating on trying to get a structure for a DNA methyltransferase. And we finally got one, and we found one where it was binding to DNA. And to everybody's amazement, the base that was going to be methylated had flipped 180 degrees right out of the helix into the active site of this enzyme. And so immediately we started to think, well, are there other things that might be base flipping? And the obvious one was to think, well, if other enzymes that do chemistry on DNA, if they do it on the bases, maybe they need to flip the base out. And sure enough, every enzyme we know of that does chemistry on DNA bases uses this base flipping process. And then something else struck us, or it struck me anyway, and that is, if you look at this structure, what's happened is that the energy that is holding these base pairs together, half of it is gone. And so if you were going to open up a helix, this would be a great first step. So you flip out one base, and then half of the stacking energy that is holding this next base in place is lost. So you could continue and open up the helix. And so I made a suggestion at the time, and it was published in Cell, that maybe RNA polymerases, DNA polymerases, maybe this is how they get into the helix and how they get everything going. And we now know, as of within the last 2 or 3 years, that this in fact looks as though it's true. That everything we know about when you're starting transcription or starting replication, you do base flipping to start opening up the helix. So that was nice to go. So now let's talk about New England Biolabs a bit, because in 1992, when I left Cold Spring Harbor to take another job, I had thought about actually starting a DNA sequencing company, because it was obvious that there were a lot of people who were going to want this, and I was kind of interested in DNA sequencing. But then when New England Biolabs, for whom I'd just been a consultant all the previous years, when they heard I wanted to leave Cold Spring Harbor, they said, well, why don't you come and work for us? And they made me an offer I couldn't refuse, and so I went up there. Now, New England Biolabs is a very interesting company in many ways. As I told you, it was started in order to make money to support research. And today we have about 550 employees, of whom a little over 100 are doing research, and that research is all paid for by the company. The bulk of it is basic research. Either it's basic research about some of the enzymes that we sell, or it's basic research about parasitology. We have a big program in filariasis. Filariasis, I don't know if any of you saw the movie The Elephant Man. It's a filarial disease, it's a worm, it gets in you. The babies try to get out of you, and when they do so, they come out through the skin, and you get these big bumps that form, and it's really ugly. And we did that because no one else was working on it, and in fact, we've spent more money on that than NIH have spent on it in the last 30 years. So, what did I do at Cold Spring, at Biolabs? So I first became a research director there. We started a program or continued a program to clone restriction enzyme genes for commercial production. Many of the strains that they were originally found in, they just don't produce a lot of them, so you can put them in E. coli. And as a result of doing that, we came across an immense number of interesting things, and we still continue to find interesting things today. In fact, just about 6 weeks ago, we found a new protein that is needed for a particular DNA methyltransferase in order to be active. And so, this was, and as soon as you find it, you look and you say, well, are there others that look like it? And of course, you go to GenBank and now there's 100 of these things. We just never knew what it was before. And so I very much like this idea of using biochemistry in order to try to characterize the products of open reading frames. We've done a lot of work in bioinformatics. I have 3 programmers who work for me, and we do a lot of genome sequencing to find restriction enzymes, but actually now more DNA methylases. I've become much more interested in DNA methyltransferases. And that's because of this technique called smart sequencing. It's from Pacific Biosciences. It's a way of sequencing DNA that is phenomenal. It really is good. What you do is you look you measure the time it takes to add the next base, and it turns out that if this base is opposite to a methylated base, the polymerase slows down, and it doesn't do it quite as quickly. And so you can measure on single molecules how long it takes to put the next base in, and when you have these gaps in time, it tells you there was a methylated base on the other strand. You can do some bioinformatics, interpret this, and in this way, it's very easy to determine methylase recognition sequences. And it's absolutely invaluable and very interesting. So what's happened as a result of the introduction of this is the discovery of methyltransferase genes, type 1 and type 3, was very low, almost none, until about 2012, which is when this technique first got started. And what's happened is the numbers have just shot way up. And we discover these things on a regular basis. They're in pretty much every bacterium. It's very rare to find a bacterium that doesn't have one or more of these genes in them. And there is no other good way to do it. If ever you wanted to find the recognition sequence for DNA methyltransferase, It's one of these projects where a new graduate student would come into the lab and I'd say, hey, why don't you work out the recognition sequence for this enzyme? They say, oh, well, that sounds interesting. And so then they go and they talk to other members in the lab and they do a little reading of the literature, and then the following day they come back and they say, hey, could you give me another project? Because it's tedious. It was very tedious work. And PacBio has made it trivial. It is now absolutely trivial to do this. But you have to save the data and you have to do a little analysis on it. Found a bunch of interesting things. I'll just mention a couple. One is, here's a thing that we found about 3 years ago now. And this is a methyltransferase that methylates. I'm looking at the two strands. So the inner red circle is one strand of the DNA. The outer red circle is another strand. And the peaks coming through here are methyl groups, areas where methylation has taken place. And what's happening is that this particular methylase will methylate one strand of the DNA, but not the other. And in Burkholderia, there's another one that does the same thing. We now know there are hundreds of these. They're all located on plasmids. And so you have to ask yourself, well, how is it that an enzyme could recognize one strand and not the other? Well, the easiest way to think about it is that if it's part of the replication machinery, then it could easily just be, say, methylating the leading strand and not doing anything to the lagging strand. And if you remember, DNA methyltransferases base flip. And so our working hypothesis, which looks as though it's correct, is that what's happening is that this methyltransferase isn't really doing methylation per se. It's derived from a methyltransferase, but what it's really doing is flipping open the helix and serving as a helicase to open up the DNA. Okay? And so this is just a repeat of that earlier slide. And so we argued if it is involved in DNA replication, then what we should be able to do is to take the origin of a plasmid and just switch it, flip it around, and do it in the other direction. And sure enough, if you do that, what happens is when it's facing in this way, it's the inner strand, the inner circle that gets methylated. When the origin is in this direction, it's the outer strand. And I can't think of any other explanation other than this is somehow involved in leading strand synthesis and likely opening up the helix. Now you might ask, why would you use a methyltransferase to do this? Well, the answer is methyltransferases are slow enzymes. They just don't do their action very quickly. And when you're a plasmid and you need to replicate, if you replicate too quickly, you clog up the recombination machinery in the cell, and the cell often will die. And so there's an advantage in doing it slowly, and we think that this is why this has happened. We've now done some purification experiments in biochemistry. You can purify the methyltransferase, and along comes the DNA polymerase when you do that. So, what have we learned so far from all of the methylation studies? One of the things that I find most intriguing is that there are an awful lot of methylases that we don't know the biological function of. So every time you see a methyltransferase, you always think, well, there's going to be a restriction enzyme associated with it. That's true maybe for about half of the methyltransferases in an average cell. But there are many cases where there are methyltransferases, they have no obvious restriction enzymes near them, and they're just solitary methyltransferases methylating DNA. Why are they there? Most of the time we don't know. There's much more variability in recognition sequences than expected, particularly for the type 1 systems. These are the systems that are ubiquitous, they're all over the place. They were the very first restriction systems ever discovered. And for a long time, people thought there were only a few specificities there. We now know there are going to be tens of thousands of specificities, and you can get these very easily thanks to PacBio sequencing. We also found for the type 2 enzymes, the type 2 methyltransferases are interesting. Type 2 enzymes are the ones you use in the lab all the time, the type 2 restriction enzymes. They're encoded by a single protein that is the endonuclease, and they have a companion methyltransferase with them. And everybody had assumed that the methyltransferase would have the same specificity as the restriction enzyme. But if you start to think about it, that's not necessarily true. Let us say I had a methylase that could recognize CCGG and a restriction enzyme that could recognize a CCGGT. The CCGG methylase would be perfect to protect. But in fact, the methyltransferase that shows up with the ACCGGT restriction enzyme also recognizes ACCGGT. That's interesting, and you might want to think about that for a little while, because it's not at all clear evolutionarily why that should be so. And I would like you to think about the following explanation, which is something that was suggested to me by a student in Novosibirsk. As a result, I was giving a video presentation from a hotel room in Japan to this conference in Novosibirsk. And at the end of it, one of the students stood up and said, is it possible that what's being selected is the methyltransferase, not the restriction enzyme? And that the restriction enzyme is just there to make sure that the specificity is maintained. Think about that for a while, because it gives you a totally new way of thinking about restriction enzymes. Oh yes, and a lot of prophages. You know, prophages, when bacteriophages infect cells, they very often insert themselves in the chromosome. A lot of these prophages carry methylases, and almost certainly because what they're doing is they're methylating the DNA to overcome restriction barriers. And there are a lot of those, many more than we'd expected. Also going to make a plug for PacBio sequencing. This is really fantastic. I love it because methylase recognition sequences are easy to find. Functional annotation of methylase genes become real easy. Closing bacterial genomes is simple. You take one PacBio run, and typically, just by computation, you can get the complete sequence of a closed genome. It's amazing. Anybody who's tried to sequence bacteria using Illumina or some of these other methods is always astounded at how well this works. And it's relatively inexpensive, and you get the methylation data on top of it. And finally, it's very accurate. The accuracy of sequencing is greatly enhanced. Only Sanger sequencing does a slightly better job. And one of the reasons is that because PacBio reads the same sequence over and over again many times, even though any single run may have 10 errors in it, they just cancel out randomly as soon as you've been around and looked at 50 of these things. With Illumina sequencing, you get specific errors. So there are some sequences that always give you the wrong answer, and we don't always know what those are. So let me tell you a little bit about myself in terms of puzzles and games. So I've always loved puzzles and games. I was on sabbatical in 1978 and learned how to play croquet on the King's College lawn in Cambridge. And I always wanted to play croquet after that. And when I won the Nobel Prize, I had the money to put a croquet lawn in front of my house. And this is me sitting in a deck chair on my croquet lawn, posing for a calendar called Stud Muffins of Science that was put out in 1997. And I became Dr. December at that time. So that was kind of fun. This is all about croquet. I also go down to the Ig Nobel Awards at Harvard. You may have heard of these. Every year they do Ig Nobel prizes. These are given to scientists who've done experiments that should never be repeated. Very interesting ones. And I love fun, so I go down there every time I possibly can. They're a lot of fun. They're now webcast. You might look for them. They're really good. And there have been a few Koreans who've won. I've also been awarded various honorary degrees by Bath, by Sheffield, and so on. I got knighted in 2008 by Prince Charles. This is Prince Charles here. This is a very fancy ceremony in Buckingham Palace. It was a lot of fun. My family thought this was great. And I've always done quite a lot of organizing of Nobel laureates to sort of for good causes of one sort and another. The current campaign that I'm on is a pro-GMO campaign. If you go to this website, supportprecisionagriculture.org, you can find out all about it. But it's basically trying to start the conversation to convince people, and in particular to convince the Green parties that this is just an issue they got wrong, And the science says these things are perfectly safe. The science says everything is fine. And the more we find out about it, the more it seems this is. And it is so important for the developed world. You don't need GMOs here, but in Africa, in South America, in a lot of the poorer parts of Asia, if they don't get GMOs and they don't improve their crops with these methods, then people are going to die. And finally, I want to tell you about the importance of luck. I think most people do not give true appreciation to luck. And so I learned this first when playing billiards. And I told you I almost got thrown out of school, but I became very good at billiards. I was playing in a match one night, and I had a lucky break. I hit the ball, and it didn't go quite where I wanted, but nevertheless, it was a scoring shot. And I took the next shot, and nothing happened. I just didn't do anything. And an old man came up to me. I was 18 at the time. An old man came up to me, and he said, "You know, you are never going to succeed well in life if you don't take advantage of luck." And so what happens is when you have a lucky break, maybe you're playing tennis and the ball hits the net and just drops to the other side. He said, if you feel guilty about this luck, you are never going to do well. When you have luck, you take advantage of it. And I think this is a very valuable lesson. You might all think about this. Everybody has luck in their lives. Everybody. I don't care who they are. And if you don't take advantage of it, but instead feel guilty about it, there was no point in having the luck in the first place. So take advantage of it when it comes your way. And you never know where it's going to show up. And here's a few others. So Kazu Kurosawa, this was this postdoc. Quite by chance, I ended up in the lab with this guy. He became the best mentor that I ever had. And I really learned how to do science from him in a way that I haven't ever learned from anybody else. He was fantastic. But sheer luck, I could easily have ended up, there were 10 other labs that I could have been put in as a starting postdoc, as a starting graduate student, but it was him. Went to Harvard instead of Wisconsin. Again, just because the guy I'd applied to, the only guy who took me, moved to Harvard at just the right time. And that was fantastic because I met so many people when I was at Harvard I would never have met if I'd been at Wisconsin. Looking for eukaryotic promoters in adenovirus. I mean, who would have guessed that that would have led to one of the big discoveries in molecular biology? This was a discovery made at a time when almost all the molecular biologies were saying, well, all the big discoveries have been made. It's only the details that need mopping up now. And of course that was not true, but this really proved it in a major way because no one had predicted anything like this. I want to tell you about this one, taking an early plane. I'm sure all of you know about 9/11 and the aeroplane that went into the Twin Towers. The first aeroplane that went into the Twin Towers, I was booked on it. Until 2 weeks before the flight, I had a seat on that plane, and I was going to a meeting over in California, and the meeting got moved one day earlier, and I had to change my flight reservation, and so I was in California when 9/11 happened. Now, if that's not luck, I don't know what is. That is probably one of the luckiest things that's ever happened to me, because if that meeting had not been moved forward, I wouldn't be here to talk about all of this. And finally, I got started studying bacterial RM systems, restriction modification systems, just because I went to a lecture at Harvard from Dan Nathans. I think but for the lecture, I would never have gotten involved in this. And as a result of that, we have made so many new discoveries along the way. It's just been fantastic. I can tell you, all I do these days is bioinformatics. I've got a computer. If I've got a good internet connection, I can do a little work from my hotel room if I wake up early before I go to bed. And I will tell you, every other day I make a discovery. I see something that people have never seen before. That is fantastic. You can't beat this. This is every researcher's dream. And all because I chose to study these systems, and not many other people have ever studied them. A lot of people, when we were first discovering these enzymes, said, oh, we're not going to do anything in that area because we can't compete with you. Well, I thought that was pretty good. But since then, we've continued, and it's been fantastic. Just an incredibly useful field of study. I just want to acknowledge the people who really have made a big effort. Richard Gelinas, the postdoc who worked with me and helped make the splicing discovery. He did about 99% of the work involved in that discovery. Louise Chow and Tom Broker came along at just the right time and did the electron microscopy, and then went on and did beautiful work characterizing splicing later on. Saida Zain was the postdoc who really provided the first evidence that the 5' end of the RNA was attached to the message but was not coded right next to it. Dan Klessig was a graduate student at the time, and he characterized fiber messenger RNA, one of the very specific adenovirus messenger RNAs. And then, of course, the very many lab members and colleagues, collaborators and friends over more than 50 years now in research. And finally, my assistant, Karen Otto, who helped me to make most of these slides. And I thank all of you for listening. Thank you.
Start the broadcast chat. This is open time for questions.
Absolutely.
Any questions?
And I might add, you can ask any questions. They can be about Donald Trump. They can be about whatever you want to talk about. Yeah. Just speak up and I'll repeat the question. I don't have to stand up. Just speak up.
My name is Koyam Ya from biotechnology, and I was supposed to be in my molecular biology class today, but you couldn't
Okay.
here. I have a question. These days there are many people talking about the fourth industrial revolution, and I am what do you think about the relationship between GMO and fourth industrial revolution, and what should young people do on this situation?
Well, I mean, there's a great deal of technology. GMOs are just one small part of the technical revolution that's taking place. If you look at what's going on in artificial intelligence, in deep learning, in the way in which computers can control things, we're going to be having autonomous cars soon. I can't wait. I just want my car to go by itself. I'll sit in the back, I'll play around on the internet, and then when it drops me off, it can go and park itself. I think that'd be fantastic. So I think there are many, many things we can do, but there are some things you have to be careful about. And so, for instance, some of the genetic modifications of humans that would be possible, if they're solving a disease, I have no problem with it, but if they're trying to make you taller or make you more beautiful, or that just strikes me as being silly. And so I think we have to be careful as a society that we regulate some of these things. GMOs, though, there's no issue. GMOs are fine, they're safe. We've been using them for 30 years. Do you have any idea of how many cattle and sheep and pigs have been eating these things for the last 30 years? Not one problem, not one instance. There'd be dead cows everywhere if the anti-GMO people were correct. So it really is nonsense as soon as you look at the science. And I think the key here in any kind of revolution is to sort of look at the science, look at humanity, get social scientists involved and really figure out what is reasonable, what isn't. The industrial revolutions, the previous ones, people have not thought so much about the consequences. Now I think we're in a position to think about the consequences. But what I see in the future is good. I like what's coming along. Thank you.
Thank you. It's our honor to have you in our university, and it's also really nice because I used to live in Southampton in England.
Oh, very good.
Thank you. It was a very nice place.
Yes, it is.
My question for
But
you...
they didn't offer me a position,
my question for you is that I'm doing my first year master program in molecular cancer biology, and I would like to know if you have any research, if you are in my position, if you have any research you would like to start on, or any subject that you are interested in.
Well, there There's so many things that you could do. So let me tell you something that I think you should avoid. Okay? One of the buzzwords floating around at the moment is something called precision medicine. In fact, we don't know enough to do precision medicine. It is the most misnamed thing I can imagine. Precision medicine would imply that if you find a mutation in a gene, you know what the biochemical and genetic consequences are. For the most part, we have no idea. You know, there are oncogenes, you find oncogenes, but you find people with mutations in oncogenes that live for very long lives because they have secondary mutations, and we don't know the interactions here. And so what I would do if I was starting all over again is I'd go back to basics and I would get involved in trying to work out what is the function of many of the genes that we're finding by sequencing. I'll give you a great example. Craig Venter recently made this minimal genome, a mycoplasma, in which he knocked out as many genes as he could. Actually, Clyde Hutchison is the guy who did it, but Venter gets the credit, unfortunately. Anyway, so they made this minimal organism, and there were about 140 genes that had to be present, that were essential for life. We have no idea what they do. I find that appalling. 140 genes, absolutely essential for life, and we don't know what they do. Let's find out what some of those things are doing. So there's a challenge for you. Okay?
Thank you.
Yep.
Hello, my name is Ji Sung-min. Thank you for your lecture. I have an interest on animal phylogeny and biodiversity. Is there any chance to the Nobel Prize takes turns on discovering biodiversity or animal phylogeny and conservation biology, which need the latest molecular biology techniques? The Nobel Prize is not the scientist's ultimate goal. Instead of discovering something new, do you have an opinion?
Well, my advice is don't do anything because you think it's going to win you a Nobel Prize. I ran a meeting in China last year called Serendipity in Science, that is chance in science. And almost every Nobel Prize winner, and there were 5 who spoke at this, what they did was to say, well, you know, I was doing this and this and this, and all of a sudden I discovered this, which was totally unrelated, and that's what they won the Nobel Prize for. I defy you to find more than 3 or 4 examples of Nobel Prize winners who were deliberately working in a field where they thought they were going to win the Nobel Prize. So you have to recognize what you want to do is find a career, find something you're passionate about, that you love to do, where you can make discoveries. And if you're lucky, maybe you'll get some recognition for it. There are plenty of other prizes other than the Nobel Prize. And if you're very, very, very lucky, maybe you'll make the odd discovery that would lead to a Nobel Prize. But if you're going to set your sights on a Nobel Prize, you're going to be disappointed. Right? I guarantee it. So don't do it. Do what you love to do because you're passionate about it, because it's fun. It's a great thing about being a scientist. You can have fun every day. It's amazing. They pay you to do it. It's great. So don't ever think in terms of trying to win a Nobel Prize. Think in terms of finding something that you love to do, that you're passionate about. Just do it.
I just have a curiosity.
Yes, curiosity is a good thing.
Thank you.
I tell you, rebellion is quite a good thing also.
First, I'd like to express my gratitude from hearing from you. It was an honor. And I'm Chunobe, majoring in biotechnology. And you told me that you were originally majored in chemistry, and you moved to a new area, molecular biology. And chemistry and biology is quite different. So weren't you any afraid moving, learning new areas in that time? Like, yes.
Well, I mean, you know, the thing to do, whatever you're doing, don't think that this is the only thing worth doing in the world. Read about other stuff too. So broaden your experience. I could have become a computer programmer. Back when we were first sequencing adenovirus, we had this guy at Stony Brook who we persuaded to write some programs, but there were a lot of simple programs that needed writing, and he wouldn't do it. He said it's too simple, it's not interesting, he didn't want to do it. And so I taught myself how to program, and wrote a lot of programs and discovered that I loved it. One night, actually one morning, I had just finished debugging the program, and I look at the clock and it's 8 o'clock in the morning. I'd worked all night and not even realized it. And about a week later, exactly the same thing happened. I was so intent on what I was doing that the whole night went by before I finished. And I decided that I was addicted, that I had to stop this, and so I stopped and hired a computer programmer. And I don't think I will be doing any more programming. But I could easily do that. I love it. I think it's fantastic. It's like a puzzle. In fact, we've just started a new technique that I thought of 2 weeks ago that requires some new programs that don't exist. And I asked one of my postdocs who's working with me to write this program and to do something. And he went about it and did it in a way that I thought was stupid. And I almost went back and started programming again. And then I suddenly remembered that you know, I get addicted to stuff very easily, so I better not do it. But always think more broadly. Think about other things that might be interesting, because there are two things. One is you may decide this is what you want to do with your life, but also you may find something that you can apply to the subject that you're doing. And an awful lot of cross-disciplinary things come because you heard about someone who was doing something. You know, you probably have read about deep learning. Do you know about deep learning? Yeah, so, you know, there are many problems that can be tackled by deep learning. Maybe there are some that would apply to you. And you could decide, well, I'm going to figure out how to do that myself, or I'll find a colleague, I'll find a friend who will do that for me, and we'll collaborate and get it done. But I think as long as you just sort of focus on whatever is your project, you don't broaden your education in the way you should. And you're constantly learning. I learn all the time. Life is, you know, science is wonderful. There's so much we don't know. It's fantastic. I'll bring the microphone over here, give these people a chance.
Hello, I'm Honik Lee from the Health and Environmental Science Department, and I'm currently studying computer science and plan to study bioinformatics in the future. And I want to ask your opinion about what kind of research will be needed in the future.
Well, you know, one of the things that is happening in biology at the moment is that we're sequencing everything that we can find. And so we are going to know about biology as a result of DNA sequencing, not as a result of doing biological experiments. And when you start to think about that, you have to think, well, what is it that we basically need to know before we can start to understand and interpret these sequences? And the answer is, well, we want to know what all the genes are doing, and we want to know where there is RNA that is coding for something interesting. And very often what you can do with bioinformatics is identify interesting things that are worth studying experimentally. And so I think this is something that's really well worth doing if you're interested in bioinformatics, to see can you find interesting problems and then either do the experimental work yourself or find a friend who can do the experimental work. But this is how we're going to find out about biology in the future. We're not going to know about biology by doing experiments, but rather by sequencing and being able to interpret those sequences. And that's a massive undertaking. I mean, this is really a big undertaking. But if we don't get started, we'll never finish.
Okay, thank you. Thank you for your kind lecture. My name is Dongbu and I'm majoring in molecular biology. And in the practical view, what is your know-how with your experiment or analyzing your data or results of your experiment?
I'm not sure I understood that.
Try
it again.
Is there any advice with the research experiment and analyze the research data?
Well, I would repeat what I said right at the very start. So think about experiments, okay? Do the experiments that seem good, and when they don't work, always be optimistic and think, well, nature's trying to tell me something, not that I screwed up. Okay? And I think failure is a good thing. This is something that so many students don't get. They do not understand how important it is to fail. And when you've got a teacher or a professor that comes along and says, oh, you're no good because you keep failing. Well, maybe you're really good and you've just chosen not to follow through and understand why your failure. Because maybe there's a big discovery being made and just waiting for you to follow through. So I love it when students come and tell me that, the last experiment failed. Wow, great, this is the best thing that happens. I think there was someone just behind you who has a question too.
Hi, I just started my postdoc in Korea. Last summer I met Dr. Rodman. He also won the Nobel Prize.
Yep.
And what he told us that science is 10% of hardworking and 90% of luck. So perhaps all Nobel winners talking about luck. My question is, during your postdoc, did you learn something from your supervisors that, like, you said to Professor Watson that you don't want to work on something that he actually hired you? So you're still thinking that if someone
when he hired me, I thought I was going to work on that. Okay. It was only later that I discovered someone was already doing it and I didn't want to do it.
So do you think it's like postdoc coming to you and say, okay, professor, I won't work anymore on this project because I think some other group's doing that. So you are angry at your postdocs or you are encouraging them?
Oh, I would encourage them because it means they've been looking to see what everybody else is doing. I mean, which is a good thing. You don't want to just get buried in your own project. But I can tell you the postdocs I like best of all, I'll tell you about one postdoc that I had, actually a Chinese postdoc. His name is Yu Zhang. He is one of the best postdocs I've ever had. He was one of these guys. He started in bioinformatics and became an experimentalist and did both. And we'd sit and talk and I'd suggest some experiments for him to do and he would go off and disappear for 2 or 3 days, 4 days. And then he'd come back and he said, I did what you asked me to do and showed me the results and it looked good. Then he said, I thought it might be good to do this too. And so then he showed me another result that he'd had from something that he'd thought up himself and did. And then I looked at it and I said, why didn't I think to tell him to do that? Those are the very best postdocs, the ones who take the initiative and do their own thing. And you can always do that. So you've got a project you're working on, always have some side project that you thought of that you want to follow. I'm very encouraging of that. I think this is terrific. And I spent my life doing that.
So do you follow your first professor that if you have great postdoc from China, you hired another postdoc from China as well?
Well, actually, I'm not quite, but I'm starting a research institute in China on the microbiome. So there will be a lot of young Chinese going there, and we're trying to set up this institute in such a way that the young people can really run with their ideas and do what they want to do without having some elderly Chinese professor telling them what to do.
So if someone is applying for a job in your lab, it's better to, like in the letter, to send information that he read about your experiments and suggest some ideas to you, or just, I don't know.
I can tell you the thing that I mostly look for when I'm hiring people is enthusiasm. If people are enthusiastic about stuff, I don't care what it is, if they're enthusiastic about stuff, they will usually work out very well. They have a passion for something. These are the people I like.
Thank you very much.
Okay.
Thank you for your lecture. You said that you changed your subject from organic chemistry to molecular biology, and I also study. I also major chemistry and interested in medicinal chemistry and molecular biology. I kind of want to change my subject like you. And here's my question: how much your knowledge you learn from organic chemistry helped you to research your new subject? I mean, molecular biology.
Yeah. So, one of the things about doing what were considered the hard sciences, like physics and chemistry and math, is that you learn how to think like a scientist. You learn how to ask questions. You learn how to design experiments that will give you good answers. And when I moved into molecular biology, most of the people doing it were biologists, and they didn't know how to ask questions, and they weren't used to getting the precise answers that an organic chemist was. And so for me, that was really good. These days, I think people who have a good handle on computation and can understand the logic of computation do very well in almost all the sciences. Learning how to ask precise questions and get precise answers is very important, and especially in biology. There's a tremendous amount of sloppy thinking in biology at the moment. People are busy gathering data. They think if they get massive data sets, they're going to understand something. For most of them, it's sort of low input, high throughput, massive output, and no understanding. And so, you know, everyone talks about big data. Everybody wants big data. What are you going to do with it? You know, if you can't understand what it all means and you're not prepared to spend the time to do it, I think you're in trouble. And this is why, you know, my feeling is if we're not going to spend time trying to understand what these sequences are telling us, why do we keep sequencing? Maybe we should stop. Okay?
Okay, thank you for your answer.
Here we have a question at the back here. Oh, you have one here. Let's do this one first and then go over there. Are you looking for an answer, yes?
Hello, my name is Lee Hyun-jin. I'm studying education, life science education. Your lecture is you're lucky is very good teacher. Why you think about Japanese professor and other teacher is good?
Why did I think they were good? Because I learned a lot from them. I mean, you know, the definition of a good teacher is someone that you learn a lot from.
She said how they can teach you what's the best aspects of them as a teacher.
Okay, well, I'm not a professional teacher myself, right? But I think if you want to teach, then you have to put yourself in the shoes of the student and explain things to the student in ways that they can understand. Okay? An awful lot of people who try to teach think that, you know, well, they have to show how smart they are and they have to use all the big words and so on. I think the key is really to put yourself in the shoes of the student and then talk to them and teach them in a way that they really can understand. So empathy, if you like. For me, that's what I like. I like someone who can put themselves in my shoes and can teach me what I need to learn. Okay? So I think your turn now, if we can get the microphone up there.
Last question.
Hello. First, I want to thank you for the wonderful speech, and I was surprised to know that you're one of the Nobel laureates who signed the letter to Greenpeace. And honestly, I have come across so many posts and websites about anti-GMO, anti-science, and also on many on the same sort. And my question is, how do you think should we react to those people? Should we just ignore them, keep doing what we are doing, and just let time prove the truth, or should we defend GMO and defend the new inventions in the name of science?
Yeah, I think the answer is the latter, that if you keep quiet, they've already won. And so it's very important, I think, for scientists always to defend science, always to try to explain science to the general public so that they will understand it. And eventually, hopefully, these people who are anti-GMO and anti-other stuff, anti-vaccination, these people are nuts, but eventually one can hope that you can train them and teach them and make them into better people. And one of the keys here is that I think you should take the grandmother test. Okay? The grandmother test says, can you go and explain to your grandmother what you do every day? Can you explain it in such a way that she will understand it and that she can then go and tell her friends accurately what it is that you do and why you're so great? And if you can do that, then I think you found the secret for communication. Okay, so often as scientists we think, well, if we can really explain the science in great detail, everybody will get it. And it's not true. You have to find ways of explaining it such that people can understand it. Let me give you one example that I give when I give my GMO talk. When you compare traditional methods of breeding plants with modern methods, with the so-called GMO method, the difference is akin to taking a GPS system, got a car with a GPS system in it, you've got a car without a GPS system in it. How do you get the GPS system into the car that doesn't have it? Do you just pick it up, transfer it and put it in the car? Or do you break the two cars apart, mix everything up, and then select the one that had the GPS system? Well, I think it's obvious. You move it. That's what precision agriculture is. That's what the GMO method is. It's very straightforward. It's the modern way of doing things precisely, and it is so much better than the traditional methods of doing things. If you go to the website, supportprecisionagriculture.org, there is a list of stuff there. One of them is videos. You go to the video section, you can see lectures that I've given on this subject. You can see slides that are available that describe this. You really have to find ways of explaining to your grandmother in words that she will understand without using too much science. Okay? Make sure the science is accurate, but don't use all the scientific terms and jargon that people don't understand.
Thank you for your answer.
Okay, you're welcome. Are you going to let him have a go?
Thank you for your lecture.
That was the question.
Yeah.
You got to do it quickly.
Yeah, many questions I was thinking to ask you, but my friends already asked them. I have one question that, can you speak a few words about the Nobel Prize sold out by, I'm not sure, Watson or Crick?
Well, actually, quite a few people sell their Nobel Prizes from time to time. In the case of Jim Watson, he sold it. A Russian oligarch bought it and then gave it back to him. That sounded like a bit of a fix to me. I'm not sure that I really believe that story. But, you know, sometimes Nobel Prize winners fall on hard times. And a good way to raise some money is to sell your prize. I know of several who've done that. Okay.
I think I worry about you higher.
It's entirely up to you.
Okay. Due to our time limit, and I think Dr. Robert has hired this more than 1 hour. And I would like to express my sincere respect and appreciation to your lecture. I think his right research and biography is good answer for your science you made today. Once again, I express